around the T cells. And the cell that controls them was discovered here in Rockefeller University Hospital, again, by Ralph Steinman, who is a couple floors down from me in the gray building over there. And what he discovered is there's a very special control, like a master control cell in the lymph node. And that's called a dendritic cell. It's this gray cell here. This is an actual picture. This shows it the best in this white arrow. The dendritic cell is this beautiful cell. It can fold around the T cell, and the T cell's here in the middle, and instruct it. And it can instruct that cell to go out and become a killer, or it can instruct that cell to chill out and not become a killer. So it's an absolutely critical regulator of whether these cells become killers or not. And if it licenses them and gives them permission, then they can go out and recognize these antigens. So to do that, actually, the dendritic cell has to have the antigen inside of it, just like a viral protein, for example. And it has to break it down and put it on its cell surface on that receptor. And that receptor has to engage the T cell. And it does it along with some extra molecules that license this to become a killer. So there's a licensing step that goes on here, but there's also the same presentation of the peptide from inside the cell to the outside. So that works fine if the dendritic cell gets infected with the flu, which it can, or a virus. But it doesn't actually work, and there's a paradox here that one of my students kept bothering me about, which was great, okay? He said, the neuronal proteins, this protein from the cerebellum that's controlling your movement, isn't made ever inside a dendritic cell. These are cells in your lymph node. So there's a paradox here, which was, how could the tumor antigen, in this case a neuronal protein that the tumor had turned on for nefarious purposes of its own, how could it get into the dendritic cell to start this licensing process? And we knew the licensing process had to work because we could see at the end of the day there were killer T cells killing the tumors in these patients. So he reformulated this sort of question, which was, there had to be a way that the tumor cell proteins get exposed or get somehow inside of a dendritic cell. So there's a real mystery. And in fact, the answer to it came from my going to a seminar, not unlike this one, in the Rockefeller Hospital also. We have a seminar series every Wednesday. There's a guy named Anthony Rosen, works with his wife, Cassiola, and they were studying an autoimmune disease called lupus. I don't know if you've heard of lupus, but lupus is a very poorly understood autoimmune disease that has, as one of its features, if you go out in the sunlight, your rash, your autoimmune disease gets much, much worse. And so he recapitulated that by taking skin cells in a dish and giving them sunlight. And these skin cells make one of the antigens, the proteins that are under attack, a lupus antigen here, inside of the cell. So this is the whole cell here, and it's actually inside the nucleus of these cells. So what he found, in fact, this antigen is very similar to some of the paraneoplastic antigens. So I was interested in this guy's seminar. What he found was, so here's the antigen inside the nucleus. After he gave them sunlight, what happened is the cells started to die from too much sunburn, basically. And the protein inside the nucleus got packaged in these membrane-bound bodies called blebs. Do you remember the movie I showed you, where those blebs came off of the suicidal tumor cell when the T cell was killing it? Well, this is a natural process whereby the same sorts of blebs got made. So we sort of put two and two together and said, maybe this process of cell suicide is critical to the immune response. And so the idea is that these blebs here may get taken up by dendritic cells and eaten inside of them. These cells are a cell that likes to eat debris. And maybe what they're doing is eating this special suicidal cell. They take it inside, and in fact, that's what happens. The red cell is a suicidal cell here, the green cell is the dendritic cell. And what the dendritic cells do, they before they get to your lymph node, they're scattered all through your body. And they're sentinels, guards. And what they're on the sniff for, they're sniffing out suicidal cells. Because when cells undergo suicide, either from sunlight or from death from a T cell, the suicide is a special signal that there's something maybe wrong. And viruses, in fact, when your cells get infected with chickenpox or measles or flu, they induce suicide. It's one of the body's defense mechanisms against something going wrong. So when they undergo suicide, these dendritic cells eat them. There it is inside the dendritic cell. Then the dendritic cell packages this, it breaks it apart. It puts all the pieces of this on the surface and runs down to the lymph node and asks, if this is dangerous, maybe you have a T cell that can recognize this and go out and kill other cells that have this viral infection or this cancer. So in fact, we can show that this works, this idea works for cancer cells. So the idea here is that we can take a cancer cell, induce suicidal death in it artificially in the laboratory, feed it to a dendritic cell. So the dendritic cells eating the cancer. And we think this is what triggers the paraneoplastic disease in these patients. And that dendritic cell having eaten the suicidal tumor, we can now inject into a mouse. So this is a mouse with a tumor here that was injected with just a dendritic cell node that hadn't eaten the tumor. And this is the growth of the tumor is really bad, okay? This is the same mouse, the same tumor, but the mouse was also injected with dendritic cells that had eaten a suicidal tumor, okay? And this can prevent the outgrowth of the tumor in the mouse. So this sort of thing is exciting. It's a way of trying to explain what we think is going on. That there's a gatekeeper phenomenon in these immune responses where suicidal death of the tumor is a gatekeeper whereby dendritic cells can recognize something's wrong in that cell, eat it, and stimulate T cells to go back and kill the tumor. Now there's a famous scientist at Harvard named Judith Vogel who discovered endothelial growth factors that make blood vessels form. Felt those were important in tumors. And for many years he was able to cure mice of cancer, but it didn't work at human. He used to sort of half joke and say, if you're a mouse with cancer, we can do great things for you, but it doesn't do any good if you're a human. So we have gone back now to the Rockefeller Hospital. We set up a system, very carefully controlled, approved by the FDA, Food and Drug Administration, to grow these dendritic cells now from cancer patients. So we take patients in the hospital, we have nurses and scientists, there's a big team working on this. So we bring a cancer patient into the hospital, draw their blood, and we can make dendritic cells from their blood, and then feed those dendritic cells tumor cells from the patient or from cell lines. Make them apoptotic, so make them suicidal. And then these dendritic cells will eat them. Okay, we can then inject these into the skin of the cancer patient, inoculate them, and basically make a cancer vaccine that's modeled on what we think is actually going right in the paraneoplastic patients. This triggering of the T cell response to kill the tumor. And we've done that in several different cancer models. We finished our first 2 studies in prostate cancer and are now trying this in brain tumors. And when you do a clinical trial like this, so we're going bedside to bench and bench back to the bedside. Now, this is this translational research. The first thing you have to do, this is a brand new treatment. So the first thing you have to do is say, is it safe? Did we trigger some autoimmune disease in the patients? And the answer is no. 24 out of 24 patients had no side effects. The second thing is maybe it was sick, but it was dead. It just didn't do anything at all. So you have to ask, did it stimulate an immune response to the tumor? And we can show all sorts of assays that it was. This is one such assay. You can see big red spot on the skin when we do repeated injections because there's a massive immune response that gets stimulated against the cancers here. And then finally you ask, is it effective? Which is something you can't do very well on a small scale. You get little hints of it. To really prove the effectiveness of something, you really have to have a placebo and a treatment group and blind yourself and ask really in an objective way that these patients get better than these patients. So that's the kind of thing we're thinking about doing now. So this is exciting for us. We can monitor the immune response with various assays that I won't go into and try and develop from this sort of Rosetta Stone of these rare paraneoplastic patients a vaccine that we could use in any cancer, okay? The last point that I'm going to make, and then I'm going to quit, is the basic science. So we can take this rare phenomenon of tumor immunity in these patients, clone